So now that we have covered all of the receptors of glutamate, let's start talking about the systems. So one of the main systems of glutamate in your brain is plasticity. Now, you've probably started to cover some of these topics of plasticity in previous courses, but now we're going to talk about the role specifically of glutamate and those NMDA receptors in plasticity. So when we're talking about plasticity, then we're mostly talking about learning and memory. So a lot of this is happening in the hippocampus. Now remember, some of this is also happening outside the hippocampus, but mainly the hippocampus is involved in learning and memory formation. So when we talk about synaptic plasticity, we're talking about the changes in strength of the synapses in response to activity. So we're changing the strength of our synapses in response to previous glutamatergic activity. Okay? So there's really two types of plasticity, or two main types. So we've got long-term potentiation, or LTP, as I'll be calling it, which is a persistent increase in strength after lots of glutamatergic activity. And then we've got long-term depression, or LTD, and this is a persistent decrease in the synaptic strength following not so much activity of our glutamatergic synapses. Okay? Now, it's important to note that while we learn a lot about LTP and LTD and its role in learning and memory, these two processes are actually only described experimentally. So in previous classes, they might have lied to you a little, saying like, this is how it all works, which this is how we think it all works, but we haven't actually been able to demonstrate that LTP and LTD are actually the fundamental processes that underline learning and memory in either humans or animals. So we think this is how it all works, but it's actually kind of just theoretical. Okay? So let's look at the plasticity in the hippocampus, where our learning and memory occurs. Now, obviously, because we associate the hippocampus with learning and memory, this is the part of the brain that's most commonly studied for this. And this is the part of the brain that's most commonly studied for LTP and LDP. This is actually a good area to study, though, because a lot of the circuits and a lot of the synapses are glutamatergic. So if we look over here, this is a rat brain or a mouse. Not really sure if there's a difference. I work with birds. But here's the brain. You can see where the hippocampus is located. So this is just a blown up slice of part of the hippocampus. You can see we sliced the hippocampus this way, and we took it out and laid it down. Okay, so this is the front, this is the back. So in our hippocampal slice preparations, so this is an ex vivo preparation, remember what that is. Then we kind of leave from the pre-afferent path to our dentate gyrus and we follow the path into CA1. Okay, so all of these circuits are intact and accessible, so we can study the entire thing. That's why we didn't cut it like up and down or anything like that. It's because we want to be able to see the entire circuit. So using these types of cuts, then this is where we first kind of theorized LTP and LTD. So let's start with LTP. LDP is pretty much just the opposite of LTP, so we're going to focus on just LTP. It's really just easier. Now, LTP occurs through coincidence detection. Remember when I mentioned that when we were talking about NMDA receptors, how they're coincidence detection devices? What? It's almost like it all connects, guys. So pretty much what happens is with a single stimulus, then we've got release of glutamate from the presynaptic cell. It enters the AMPA receptor, and it's all good and well, but because we only get like a little bit, then not enough sodium enters the postsynaptic cell for there to be a postsynaptic membrane depolarization. So because there's no depolarization, even though we've got glutamate and glycine attached to our NMDA receptor, the magnesium block is still there. So the NMDA receptor is closed. However, during what we call tetanus or repeated stimulation, then we get lots and lots of glutamate released into the presynaptic or the synapse, and we get a lot of passage of sodium into the postsynaptic cell. Okay, so really what's happening is there's probably multiple AMPA receptors open now instead of just a few. And because enough AMPA receptors are open, we get enough sodium influx into the cell that it causes a postsynaptic membrane depolarization, which causes the magnesium to be expelled from the channel, allowing sodium and calcium into the postsynaptic cell. So that's all well and good. Now, because this is happening, then it can lead to LTP. Okay, so this is not LTP. This is what will lead to LTP. Now, you also probably might be asking yourself, well, Erin, you also said we were going to talk about this CAMK2. What we are. So, CAMK2 is our calcium calmodulin dependent protein kinase 2. This is a picture of what it kind of looks like. And this is localized, so it's kind of like buddy system with the NMDA receptors. So it's on the intracellular face, so it's inside the postsynaptic cell, kind of hanging on to that NMDA receptor. And what it does is it phosphorylates numerous cellular targets and actually initiates the early phase of LTP. So it helps in plasticity. So if CAMK2 is involved in early LTP, then we should probably talk about that. So how it works. We've got our little diagram over here. We're going to be very familiar with this diagram. So we've got our AMPA receptors in blue, NMDA receptor in red. So once the NMDA receptor opens, then not only does sodium flood in, but calcium also enters the postsynaptic cell, right? So the calcium is going to actually interact with our CAMK2. Now here we just have CAMK2 kind of like hanging out here, but it's actually going to be like a protein like right up here, right next to the NMDA receptor. It was crowded. So calcium enters through the NMDA receptor. It interacts with CAMK2. CAMK2 is then going to send some of its power. I call it power. It's like magic power, you know. And it's going to phosphorylate our existing AMPA receptors. Now, when you phosphorylate these receptors, what it does is it increases the availability of the receptors. In other words, sometimes, or in normal situations, then not all receptors are available at the same time. They kind of take turns. But when you phosphorylate the AMPA receptors, then you're increasing the amount of available receptors. So instead of having like 3 shifts of receptors and only 1 shift is available at a time, now all shifts are working at the same time. Okay? So now we get more sodium into the cell, which will increase the possibility of an action potential quicker. Now, as CAMK2 is phosphorylating our AMPA receptors, it's also going to do a signaling cascade to stuff that we don't care about and increase the amount of AMPA receptors that are inserted into the synapse. So that means we're now going to have more AMPA receptors, and these AMPA receptors, because it's a whole cascade thing happening, are probably also going to get phosphorylated, which means they're going to be available as well. So now we have more AMPA receptors available, all allowing sodium into the cell. This is all allowing the postsynaptic cell to be depolarized quicker. Okay? Now, as if that wasn't enough, calcium doesn't just attach itself to CAMK2 and work on the AMPA receptors. Calcium will also go and activate some retrograde messenger signals, which will leave the postsynaptic cell, go to the presynaptic cell, and say, yo, dude, release some more glutamate. And it will. So now we have extra glutamate being released from the presynaptic cell, which can now bind to these brand new AMPA receptors we have. So now we have extra glutamate, we have extra AMPA receptors, more AMPA receptors are active, therefore increasing the amount of positive ions into the presynaptic cell. This means that we get a quicker depolarization of our postsynaptic cell, therefore increasing the rate of action potentials. Okay? So that's early phase. What could possibly be late phase? So in late phase LTP, then our activation of CAMK2 and PLC coverage on another signaling kinase, ERK, or extracellular signal regulated kinase. Okay, so for this, you're going to want to know what CAMK2 is and ERK. We're not really covering PLC. You can look that up if you want to know. So, but what is ERK? So ERK triggers downstream changes, including phosphorylation of transcription factors. So transcription factors obviously are factors that are involved in transcription. So that is that gene synthesis or reading of the genes to produce proteins. Okay, so gene synthesis is induced, increasing the production of AMPA receptors. So now, not only are we moving the AMPA receptors we already have in storage to the synapse, like we did in early phase LTP, now we are producing more AMPA receptors, okay? Because we used all the ones in storage, but we still want more. And that's what ERK does, is it tells the transcription factors, yo, we need more. It gets the genes to synthesize itself into proteins and make these AMPA receptors. So the synthesis processes are very important for long-term maintenance of potentiation, because if you don't make more AMPA receptors, then eventually we're going to lose the AMPA receptor, the influx of AMPA receptors on that synapse. Because remember, eventually everything dies, including receptors. Now, usually we've got AMPA receptors in storage to replace the ones that decommission. But because we put the ones in storage on the synapse, now we've got nothing in the storage room for the ones that break or are decommissioned. So we need to make more. So if we continue to make more AMPA receptors, then we can continue to replace the broken old receptors as they go out of business. So we can maintain the influx of AMPA receptors on the synapse, therefore maintaining our long-term potentiation. Okay, so you can see how the NMDA receptors are very pivotal in LTP, how they kind of kick everything off because they allow calcium in, which can then activate CAMK2. Now, obviously this means that LTP induction depends critically on the NMDA receptors. So NMDAR overexpression actually increases learning in mice. So if you have more NMDA receptors in mice, then we actually see an increase in learning. So if we kind of look at this graph over here, wild type, so the blue line, that just means a normal mouse that has a normal level of NMDA receptors. And then we got Doogie 1 and Doogie 2. And so these are mice that have an overexpression of the NMDAR gene. Okay? So these mice were engineered to overexpress this subunit. They're just called Doogie mice. And what we see is that they have an increased retention in novel object recognition tasks. So if we look on the x-axis, we've got 1 hour, 1 day, 3 days, 1 week. And in our blue wild type mouse, you can see at 1 hour, retention is pretty high. Their exploratory performance is about 70. And then as the week goes on, it lowers to about 50%. However, in our Doogie mice, then our exploratory preferences stay pretty high. They stay at about 70 for a whole day and then drop to 60 before at a week they go down to about 50%. So this really just means that the rats are more interested in exploring and learning the specifics about the novel task instead of just kind of seeing it and being like, yeah, okay. So they have this prolonged learning, right? So this LTP. So that's where we're going to stop for plasticity. Then for the rest of this, then we're going to talk about cell death pretty much. And toxins to the body using glutamate.